Hello. Today we are going to be working on a protocol. Here's the protocol, cloning from cDNA. And what we're going to be doing is starting the process of amplifying a fragment of a gene of interest and placing it in a vector that we can use for making double-stranded RNA and riboprobes. So what this looks like is kind of diagrammed here. So we know the sequence of genes that might interest us. So this might be a gene that you have a hypothesis about having a role in regeneration. We design primers that are specific for that mRNA, so little small primers. We're going to use those primers in a cloning PCR to amplify a piece of the gene of interest. Then we're going to digest this PJC53.2 vector, which normally has some other components in it. We're going to digest it and then ligate it with our amplified portion of our gene of interest to create a plasmid like this. So this plasmid will be PJC53.2 in the backbone. It will have a gene of interest, and it will have these T7, T3, and SP6 promoters. These are going to be important for making double-stranded RNA for RNAi and for making riboprobes, which will be important for a lot of what we do in the lab. So the first step for this is to set up this cloning PCR. A PCR, or polymerase chain reaction, is a reaction in which you combine template DNA. This says mRNA, but it's actually cDNA, which is synthesized from the mRNA that we're going to use as template. We are going to use a buffer, dNTPs, which are the building blocks of DNA, and these little gene-specific primers, and an enzyme called platinum Taq, and that's the enzyme that will be used to amplify this component, or this insert for our vector. And platinum Taq is a DNA polymerase. We use this platinum Taq instead of a regular Taq because it is higher fidelity, meaning it will make fewer mistakes than regular Taq, and you can look up more about platinum Taq if you'd like. So this is the protocol. The first thing we're going to do is set up a PCR, which if you've started with different processes in the lab, you might have already done this. We have two different PCR protocols, this cloning protocol and a hot start protocol, so just make sure you understand that those are both conceptually similar and that you amplify a piece of DNA from a template, but the reaction components in the reaction are somewhat different. So I've already filled out my lab notebook page. I'm going to clone 4 inserts, so I have them kind of numbered here. When you figure out what genes you're going to clone, you'll be given primer pairs, so forward and reverse primers that will bind to the template to amplify a piece of your gene of interest. You may design these yourself. And so you'll have a forward and a reverse primer. You may be given an empty tube that looks like this, and in that case it will probably have a dried up or kind of desiccated version of that primer in it. What I do to start with is I spin these empty tubes down to make sure all the primer is on the bottom of the tube, and then add TE, which is tris and EDTA. You can look up the recipe online if you want, so that this will be a 100 micromolar stock. From that 100 micromolar stock, that's kind of your backup stock, you're going to make a 10 micromolar working stock, and that's what you'll need for the reaction. So I have my primer pairs in a bunch of these tubes. I've already added my TE to get them resuspending, and I'm just going to give them a little shake. Alternatively, you might be given a primer that's already been resuspended. Either we purchased it that way or someone else has already done that for you. But your starting point will need to be the 10 micromolar forward and reverse primers. The other components of the reaction should be provided for you, so that will be this 10x PCR buffer. All enzymes work at a specific pH and in a specific environment, so that's what the buffer is for. These dNTPs, which are the building blocks. The magnesium chloride, which is an important factor for this enzyme working. This is your forward and reverse primer. Every once in a while we'll resuspend these together, so you might be given something that has both of these in it, and you would be instructed to use only 1 microliter probably. cDNA, which is just mRNA reverse transcribed into DNA using reverse transcriptase. And so that basically is a mix of all of the different mRNAs from the animal converted into DNA form. And then the platinum tech, which is the enzyme. So this is 225 microliters with sterile water. So I've already written this out. I'm going to make a master mix in just a moment. So you can first start out by just making sure you've read over the protocol, that you know the components that will be needed, that you know your genes, and that you've set up this reaction and done the math to figure out exactly how to do it for your number of samples. Okay, so at this point I've set up, I have my 100 micromolar primer, so forward and reverse for each gene. I've made my 10 micromolar dilutions, or my working stocks of each one. And I've also labeled a tube that says MM. That's for my master mix. And further, I've gotten out some PCR tubes that look like this. They're a little bit smaller. I have a little rack in the lids that look like this. And this is where I will set up my PCRs. So this protocol will tell you how much of everything you need to add. But because I'm setting up reactions with 4 primer pairs, and often you'll set up many reactions at the same time, what I've done is create a list, a master mix. So this is just what it would take to set up 1 reaction. I'm setting up 4, so I'm going to multiply this by 5 and then add these things all together in my master mix tube. You'll notice that I did not include my forward and reverse primer. Those are going to be specific for each reaction. So the first gene you clone, these primers will go in the first tube, and the second, and so on. I've also numbered my genes of interest that I'm going to clone. It's hard sometimes to write the whole thing on all of your tubes, so if you're going to number things, just be really careful to label things and then also initial and date your tube so that you know what's what. Okay, so the next thing I'm going to do now that I've set up my primers and my master mix tube is to go get these reagents. I have some sterile water. I'm going to go get the 10x PCR buffer, 10 millimolar DNTPs, 50 millimolar MgCl2, the cDNA. And then the platinum tech is an enzyme, so every time you set up an enzymatic reaction in this lab, you're going to only get that out right when you need it. So you'll get everything else out, thaw them, combine them in the master mix, and then only get out the platinum tech at that time. There's a lot of common stock reagents in the freezer, in the far bay of the lab. And so if you check out the lab tour, I'll kind of show you where that is. But the first time you set things up, you can also just ask. So I'm going to get all of the buffer components and all the things that need to thaw out and let them thaw. And then I will show you how to combine those into a master mix and then grab the enzyme for the last component. Alright, so I've now thawed out all of my reagents to set up the master mix for my cloning PCR. First I have this 10x PCR buffer. I have 10 millimolar DNTPs. I have 50 millimolar magnesium chloride, and just be careful with this, there's also 35 millimolar that we use for another PCR that we tend to do in the lab. I have all of my primers, which have been diluted to 10 micromolar. I have my cDNA. This is some cDNA that I made, but there are also tubes that our technician has made for the lab to use. And then I have a tube of sterile water. So I have my master mix tube just labeled MM, and I'm going to combine all of these reagents into the tubes. And remember, I've calculated what I need to set up 5 reactions, but you might have 10 reactions or 20 reactions or just 1 reaction. So you may have a different need to add different amounts to your reactions. I would say I usually set up a master mix when I have more than 2 tubes. If you just had one of these reactions, you can skip the master mix. Master mixes are meant to prevent you from spending a ton of time and tips and everything pipetting something a bunch of times. And the fewer times you pipette, the less likely you'll make an error in pipetting. So for both of those reasons, we tend to set up a master mix when possible. So I'm going to first start out with my water. So this is just sterile water. I'm using just the regular tips, not the RNase-free filter tips for this protocol, and I'll use those for the entirety of cloning. We're only dealing with DNA, so we don't need to use the RNase-free tips. So I'm going to add my water to my master mix. Next, I'm going to add my PCR buffer. I usually add the water first. That's usually the largest volume. And that way I'm adding everything else into the water. So I find that to be a little bit easier. You also want to make sure everything's completely thawed before you add it. You don't want to be in a hurry and only partially thaw things, because if you've only partially thawed a tube, it may not be fully resuspended and mixed up, and so you might be pipetting something that's not exactly the concentration you expect. And then after the PCR buffer, I'm going to add my TNTPs. I also like to put things in order. You can't see that on my rack, but I have everything in the same order I'm going to add it. That way I know which tube I've added, even if I'm talking or something like that. You can also put them in a row and then move them back a spot once you've added them, so that you don't forget where you are if you get interrupted or something like that. Now I'm going to add the magnesium chloride. Remember, magnesium is important for the function of many enzymes, so that's why we're adding it. And then I'm going to add the cDNA. Anytime you're pipetting nucleic acids like DNA or RNA, you don't want to pipette them up and down like this, because that can shear the DNA. So once you put all those components together, you're just going to flick your tube to mix. And the last component we're going to add is the platinum tack, which is the polymerase. So I'm going to go to the freezer and pull that out on a little cold block, and I will show it to you when I get back. So I've retrieved our platinum tack. Looks like this. And I have kept it on a cold block that looks like this. So anytime you pull out an enzyme for your molecular biology work, you want to keep it cold. Enzymes are proteins, and proteins can be degraded if they're heated and thawed and warmed and cooled multiple times. So we keep these enzymes very cold all of the time. They stay in the freezer, and then anytime we get them out, we leave them on this cold block. You'll notice though, if you look at it, that this is not frozen. So you can see that there. And that's because most enzymes are kept in a mixture that contains glycerol, which has a colder freezing temperature than water. And that glycerol will stabilize the enzymes at cold temperatures so that you don't get a freezing and thawing of the protein, which can also be very damaging. So when you go to pipette this, you might notice that it's a little bit viscous or kind of thick in consistency. So just be really careful to look at what you're doing when you're pipetting to make sure that you get the right volume. And I just have like some little tricks like this, like holding the lid in my hand. I'm going to look to see what I'm doing to make sure that I get 1 microliter in my tip. Put the lid on, put it back on the cold block, and then I'll add my 1 microliter, which is what I need of my platinum tack. And again, I'm going to just mix this really well. And before I do anything else with my master mix, I'm going to take my enzyme and put it back in the freezer block. This prevents things from happening like, you forget about it and then you leave it out on your bench. So as soon as I use the enzyme, I put it back immediately. Okay, now that I'm back, I have my master mix completed. And according to my protocol, I need to add 23 microliters of that master mix to each one of my PCR tubes, which again, I have already pre-labeled in a little PCR tube rack. So I'm going to add my master mix, just using 1 tip over and over since it's the same solution to each of my 4 tubes. Again, you may have a different number depending on how many things you're trying to clone. We always want to put our tips, I should have mentioned, into this little tip container. Those should not be thrown in the trash because they can puncture the trash can, but tubes and extra spent reagents can just go in the trash. So now I have my master mix of my PCR components, and what I need to add now are my primers. And so for each one of these 4 reactions, I need to add 1 each forward and reverse primer. So I'll show you the first one and then I'll turn the video off while I do the other one. So this is my sample 1 forward primer. I'm going to just take 1 microliter of it and add it to my first tube. Using a second tip, a clean tip, I'm going to add 1 microliter of the reverse primer into the same PCR tube. So now in my first tube I have the reaction mix in each primer. I'm going to stop the video, but I'm going to do the same thing with my second, third, and fourth tube, and then we'll cap the tube and go start the reaction. So now that I have finished this, I've added a cap. So this just has the reaction mixture plus 1 microliter each of the forward and reverse primers. I put a lid on these tubes, and then I've used my wrist to kind of flick them down so that if there were any bubbles or anything, at least the solutions are at the bottom. And then I'm going to go put these in the PCR machine, and I will take you with me. My hair is doing something crazy today, so sorry about that. So we have 3 PCR machines in the lab. That's these 3. There's 1 control panel. You want to use the smallest PCR block for the number of samples you have. So this has 32, this has 64, and this has 96. I only have 4 tubes, so I'm going to use this smaller space. I'm going to put my tubes in it and pull down this arm to keep the lid right on my tubes. Next on the panel, I'm going to use the arrows to select PCR from cDNA. I'm going to hit start. It's going to give me a choice of which block I want to use, and I'm going to use the 32 tube block, and then it will start. And what you should see is that it will say running, and then this light will also start blinking up here. So this takes a few hours. You could set this up at the end of the day, and the end of the program will be a cool temperature, so it'll basically put itself in the refrigerator after it's done. Alternatively, you can go ahead and work on the next components of the protocol. So I'm going to do that in a moment. So the next steps will be to digest this PJC vector. I should mention that after you're finished setting up your PCR, you've already put your enzyme away, but you also need to put all of your other reagents away. And that includes your primers. Those can all be stored at -20 degrees in the freezer. The second part of the protocol, and again we're doing this cloning from cDNA, the second part of this protocol is a vector digest. And so what we're going to do is enzymatically digest a vector to prepare it for ligation of our PCR products into it. If you look at the protocol, it will show you that this is the vector we're going to use, PJC 53.2. This was generated in my postdoc lab. And it has a backbone that has ampicillin resistance and kanamycin resistance, which is what we'll use to select for the plasmids. It also has, you can see, some promoters, so T7, T3 up here, and T7, SP6 down here. And then it has 2 genes between those promoters, so it has CAM resistance, which is kanamycin resistance, and CCDB. And CCDB is a toxin for the bacteria that we use for our transformation. So if you just tried to transform this plasmid into our bacteria, it would kill the bacteria because this plasmid would cause them to generate this toxin. We can propagate this plasmid in a bacteria that's resistant to that toxin. But our regular E. coli, this plasmid would kill them. So what we're going to do is digest this vector using these 2 sites. Here it says ADH1. It's in the vector twice. ADH1 is an isoschizmer with EAM1105I. So we're actually going to use that enzyme. What that enzyme will do is cut the plasmid at this ADH1 site and at this ADH1 site. And it will leave ends that can be ligated with an overhang that's present on the ends of our PCR product. So basically this vector is the perfect recipient of the PCR product without having to do anything else to the ends of that product. Once we have that digested vector, what we'll see when we run it on a gel is we'll see 1 long piece of DNA and 1 short piece of DNA. That means the vector has been properly digested. And then we don't even have to digest, or we don't even have to remove this insert. We can just ligate a mixture of these 2 components and our PCR product in a ratio that's favorable, And our PCR product will be ligated into this backbone. And so what that will then give us is this vector that we've been talking about, where we have our gene of interest in this PJC backbone with these promoters on the end. These promoters will be really helpful because we can use the T7 promoter to generate double-stranded RNA, or T3 or SP6 to generate a single-stranded antisense riboprobe for in situ hybridization. So the first step we have to make, besides setting up our PCRs, is to digest this vector. So we're going to incubate this vector, 1 microgram of the vector, which we have some aliquots of. It will tell you what the concentration is, so you can calculate how many microliters you need to use. We're going to use 1 microliter of this enzyme. Since that's the enzyme, we're going to add it last. And then we're going to add fast digest buffer, which has a little purple lid and looks like this. And the buffer is just to put the enzyme in the right environment chemically for it to work well. So in my lab notebook page, I've already written out how much of everything I need to add. Because my PJC 53.2 is 10 nanograms per microliter, I need to add 10 microliters for it to be 1 microgram. I'm going to add these components all in a reaction. And this time I'm only setting up 2 of these, so I've set up my PJC digest tubes. And because I don't have very many, I'm not going to make a master mix. You could do that if you wanted to. And I'm going to add them in the order of water, plasmid, digest buffer, and then the enzyme last. And just as with before in the PCR, I'm going to go get the enzyme right when I need it. So the first thing I need to do is just add the water. And these are just 2 empty tubes that I've labeled for these reactions. Again, I'm using non-RNase-free tips, just regular pipette tips. The second thing I'm going to add is the PJC 53.2, and I'm going to just use 1 tip for both of those too. Even though I'm pipetting into water, it's sterile water in a sterile tube, so it's okay to cross-contaminate that in my DNA. It's just sterile water. But then after that, I'm only going to add things using 1 tip. So the next thing I need to add is 2 microliters of fast digest buffer into each tube. And this fast digest buffer is in the same place as the enzyme in the cold block, but I got it out a moment early so that it would be thawed. Again, you always want to make sure buffers and frozen components are fully thawed before you add them to your reactions. Okay, and then the last thing I need to add is the enzyme, so I'm going to get it on the cold block, and we'll be right back. So I've retrieved the EAM 1105I. It's in a purple lidded tube. Remember, as with the polymerase, this is not frozen, it's in glycerol, and we want to keep the enzyme on the cold block the entire time. From the time we get it out of the freezer, we want to use it quickly and then put it back in the freezer. So I need only to add 1 microliter of this enzyme to each reaction. This enzyme is going to cut the vector in those 2 places, releasing that toxic insert and preparing the vector to accept the PCR products. I'm going to go take the enzyme back to the freezer, and we'll be right back. So the next thing we need to do is take these 2 digest tubes and put them in either a water bath or a block incubator at 37 degrees. So I just turned this on. It's not quite at 37 degrees yet, but we do have these 2 water baths. This one is often set at 37 degrees. You can check to make sure it's on before you start if you think of it. That way you'll save a little time. So I'm going to start a timer. This reaction needs to go for at least 1 hour. Since I started the water bath, I'm going to let it go a little over an hour, and then we will proceed with the plasmid. The step after this, as it says in the protocol, is to both quantify and run this DNA out on a gel, which we will do. There's a separate video showing you how to make an agarose gel and pour it and run it out. And You can also clean up this digest with a cleaning concentrate kit, which there's also a video of elsewhere on the site. So I will probably come back after I have run out the gel and show you what it looks like so that you can see how to move forward. What I tend to do, if I can time it properly, is to run out the gel for both the PCR products that we've generated and the digest. And that way on the same gel you can see whether your PCRs worked, how much product you got, and also ensure that the digest of your PJC worked by seeing those two bands. And then with that information, you're prepared to set up the ligation, which will be the next step of this protocol. So my PCRs are finished. My digest's finished after I let them go about an hour and 15 minutes since the incubator or the water bath wasn't on when I started. And so now I'm going to run my gel. And as I mentioned, there's another video about how to pour and run an agarose gel that you should check out. But I wanted to let you know how I set this up. So I'm going to combine for my PCRs. You may not get a whole lot of product, so you're amplifying something from cDNA, not from a plasmid like we do with the hot start PCR, so you may not get a whole lot of product. So what I'd like you to do is, for those samples, combine 6 microliters of water, 3 microliters of your PCR product, and 1 microliter of the orange G loading dye. For the vector, you can combine 8 microliters of water, 1 microliter of orange G, and 1 microliter of the digest, and that should be just fine. So I'm going to combine that all, run my gel, and then I'll show you a little bit what that looks like and how I interpret it to get ready for the ligation. So I have run out the PCR reactions that I set up from cDNA and also my plasmid digests. You can see in these 4 lanes I have 4 PCR products and nice bright bands for each product. Sometimes you'll see a fainter band, and that's okay. You can often still clone with that. And you can see here for the plasmid, you have 2 bright bands representing the 2 pieces of the plasmid that have been digested with the EAM. So that looks pretty good. We'll move forward with the ligations tomorrow.